Welcome back to the course in Nuclear Medicine Physics. Today we're taking a deep dive into radiopharmaceuticals. Now, radiopharmaceuticals are chemicals that are used to image organs and biological processes. We'll look at some ideal properties of radiopharmaceuticals such that they can be safely used in a clinical environment. We'll then look at the mechanisms of localization of radiopharmaceuticals in the body. Where do they go? Why do we want them to go there? And what can we do once they're actually there? We'll look at a few particular examples of radiopharmaceuticals that are used for both diagnosis and treatment of patients. And we'll also look at a few essential properties. I hope you enjoy this video, and let's get to it.
Okay, so this lecture will be about radiopharmaceuticals. It's a very, very important topic. This really grounds us as to the amazing applications of nuclear medicine, both for imaging and therapy. And that really comes down to the core point is that there is a very diverse range of radiopharmaceuticals for many, many, many different applications, clinical and otherwise. And so it's very important for us to understand and be excited by the range of applications and by the versatility of this field. So let's without further ado, let's get through that. I do want to acknowledge a number of individuals. I especially want to acknowledge, first and foremost, Dr. Stuart Jackson, who has been very gracious. He was a person who he continues to be a great friend to both me and Carlos, and he used to teach to our residents, the material of nuclear medicine. He's been very gracious to share many of his slides that will be used in this course for different lectures. I also want to acknowledge for this particular discussion today, Drs. Paul Schaefer, Dean Wong, Martin Lloyd, and Sarah Harsini. Okay, so radiopharmaceuticals, essentially in nuclear medicine, we use special chemicals, which is what they are, to image the structure and especially the function of organs and biological processes. Radiopharmaceuticals are sometimes called radiotracers and are labeled with radioactive compounds, which allow them to be imaged. So that's the imaging side of things. But sometimes because they concentrate in specific locations, they can be injected, they can be given at very high doses to actually treat those areas in which they're concentrating. And we will learn about this more today. But in that case, we don't call them radiotracers because the expression tracer refers to small amounts. And the idea is for imaging, we're giving small amounts, so we're not disturbing the system, where actually in therapy, your intention in a sense is to disturb the system and to attack, for example, tumors. So they are designed to mimic a natural physiological process and are tagged with a radionuclide, which emits, for example, gamma rays. So this is for imaging, so that the distribution within the body can be imaged. We will talk later about those that are tagged with radionuclides that emit other things, such as alpha and beta particles, and that is for therapy. So this particular slide is really for imaging radiopharmaceuticals. And they produce images of function, primarily as opposed to anatomy. This is why, again, the field of nuclear medicine is so exciting, because you're not just doing anatomical imaging, such as in conventional CT or conventional MRI, but you're actually looking at the function, and you're having, for example, a molecular target that you're tracking. And this can be visualized in a single image or in a sequence of dynamic images to show both static and dynamic processes. So again, you can focus on a particular organ, you can track it over time. And so a radiopharmaceutical has the following ideal properties. Again, we're talking about imaging radiopharmaceuticals here, that they produce gamma rays with energies between 100 to 300 kilo electron volts. And the reason is that we don't want energies to be too low or too high is because if they're too low, as you will learn in a follow-up lecture by Carlos, if the energies emitted are too low, the body tends to absorb and to scatter, a lot of that. So a lot of it won't make it properly to the detectors. So you don't want the energy to be too low, and you don't want it to be too high, because if the energies are too high, then you really have to build detectors that are having to stop really highly energetic photons. So that ends up making your camera more expensive. In fact, as we will learn in later in PET imaging, we're having the energy 511 keV, and that has been a challenge with PET because you're having highly energetic photons, gamma rays. So you have to build these more expensive cameras, and therefore PET imaging, this is the primary reason PET imaging is more expensive than other forms of nuclear medicine. So single photon imaging, meaning gamma cameras, SPECT imaging. But it turns out, as you will see later in PET imaging, because of the dual photon nature of PET, which is very special, you actually get superb sensitivity. So even though they end up being very expensive, but the sensitivity kind of takes you to a whole different domain. An ideal radiopharmaceutical should have a relatively short half-life, not way too short because then you don't have enough time, and not too long because then, what's the point of it staying in your body long after the imaging has been done? So you don't want it to have too long a half-life either. Otherwise, you cannot inject too much into the patient because of the harms that it has because it's staying in the body for too long. Have little or no particulate emissions, again, unless this is used for therapy. So you don't want particles to be emitted unless used for therapy. And it should rapidly localize in the organ or tissue of interest, should be non-toxic, readily available. So technetium actually kind of fulfills all these requirements. And that is why it's the most commonly used radionuclide attached to a radiopharmaceutical. And we've been learning about that, right? And so there are different mechanisms of localization. Why does a radiopharmaceutical accumulate in a certain place in the body? There's many different mechanisms by which that happens. So you sort of see this list, and we mentioned this in chapter 5 of our text as well. So you could have a range of things, right? So you could have like just a standard, diffusion, right? Or you could have cell sequestration. I'll talk about this in a second. Phosphorylation, I'll talk about this also today. And these last two also we'll talk about more today. But there are many, many different mechanisms by which this happens. So just to show you the amazing versatility and weirdness of nuclear medicine, that you could do so many things, here's an example of a procedure that has been done historically in nuclear medicine, just to see the kind of weird and amazing things you could do. So imaging the spleen, okay? So what does the spleen do? The spleen, actually, you can think of it, one of the main functions of the spleen is to filter red blood cells as it goes through it. If the red blood cells are damaged, then the spleen, does cell sequestration and it sort of removes it from circulation. Well, imagine if you take red blood cells, out of the body, and then you heat damage them, and then you re-inject into the patient. Well, once it ends up in the spleen starts to deal with it, right? And so the spleen starts to stops it from further flow. And it actually, therefore, you will see this kind of lighting up that is happening because, again, the spleen there's cell sequestration happening by the spleen because these cells have been damaged. So this is a very smart way of, for example, imaging the spleen. Now, this is not commonly done these days, but it's just showing you the excitement, especially, in the middle of the previous century when nuclear medicine was being essentially born with a lot of different applications that many of these kind of exciting things started to happen. So again, we've talked about technetium before. It does have a really nice energy, 140 keV, does have a relatively short half-life of 6 hours. It's readily available because you have molybdenum 99, which you could put in a generator. And therefore, that's why it's very commonly used. So, Carlos talked about these, and there's really three different mechanisms that, he talked about, and the text also in chapter 4, we talk about of how radionuclides are produced. One is by chemical separation of fission, for example. This is one example by which molybdenum 99 can be created. Another one, and this is through a reactor, of course. Another approach, which is again also made possible by a nuclear reactor, is to bombard a target with the neutrons from a reactor. And this is another way of producing molybdenum-99. So 2 different ways of doing molybdenum-99. This is the more common way that is produced in reactors right now. And there's a third way, and that is through cyclotrons, which we learned about last time, when you have a highly charged particle. So again, molybdenum-99 tends to be produced through the fission process. That's the most common approach that is being used worldwide right now. And again, you put that in a generator, and Carlos talked about that. But there's another alternative to that, because there's been shortages of molybdenum-99 from reactors, and that has resulted in some alerts in different parts of the world. So people have thought about, what do we do? Of course, one approach is to build more reactors, but another one is to be more innovative and to try other approaches. So actually, in Canada, and people at our very own scientists at that work very directly with us at Triumph and at BC Cancer, they came up with an idea of using a cyclotron, or they really explored that deeply, to use a cyclotron instead to produce technetium-99m. So this is Dr. Francois Baynard, this is Dr. Paul Schaefer, and this is Dr. Tom Ruth, amazing scientists at Triumph, UBC, and BC Cancer. And so here's many different ways by which potentially you could get technetium-99m through a cyclotron. So especially look at the first line here, where if you have molybdenum-100 and then use that, and you have a cyclotron that has the proton target molybdenum-100, you actually end up with technetium-99m. This is one way to do it. There's actually multiple ways of doing it, either ending up directly with technetium-99m or with molybdenum-99. But again, this is an alternative to using reactors, and they can use cyclotrons because many different sites have their own cyclotrons, for example. So this is a potential alternative approach that created a lot of excitement, at least in Canada, at least in our center, about 5 years ago, and it's been ongoing. So Carlos talked about the generator. You guys can read more details here, but he also talked about it. I won't go through details of how a generator works. So these are just examples of different radionuclides that are being used in nuclear medicine. So some of them produce gamma rays directly. So again, these would be imaged using a gamma camera, planar imaging or SPECT imaging, which we're going to learn about in detail later. Or some of them produce positrons first, and then those positrons end up producing gamma rays. And so these will end up being used in PET, positron emission tomography. But you can sort of see that different isotopes, again, technetium is shown here, but there's a whole range of them here. And you can sort of see the range of energies that are being used. You can see that in PET, we have 511 keV, but the rest of them are less than that. So for example, 140 keV, that's an important number to remember. Different kinds of half-lives, right? And different decay modes and different production methods. And also look at these last two, for example. These are actually gases, and we're going to talk about this right now. So let's say you want to study ventilation, which is, here's an example of this thing called technigas, and the idea is you have a dispersion of technetium-99m labeled carbon nanostructures, or instead of that, you could use these two other gases, inert gases like xenon-133 or krypton-85 gas, but these are radioactive gases. These can be used, for example, to study ventilation. So remember, ventilation is the study of flow of air, right? So you can study the lungs, how ventilation happens in it, while you can use another radio tracer, such as MAA, to study flow of blood. So that's perfusion. So this is a very important examination. They call it, VQ, ventilation perfusion exam. So you study the flow of air and the flow of blood. Okay, and so that actually is a very important exam. Allows clinicians to study, for example, pulmonary embolism.
So sorry, man. There was a question in the chat asking exactly this: why Cesium-133 half-life is so short, but it is using nuclear medicine.
Yeah.
Why they use it? This slide, I think, answered that question. Did it answer the question, or you want?
I think that's a pretty good question. I think because early on we said we don't want the half-lives to be too short, but on some radio tracers, half-lives are actually quite short. Like for example, oxygen 15 is another example, right? And actually, this is days, so this is not short, but this is short, right? So you've got very short half-lives sometimes. And so when we say ideally we don't want it to be too short, that doesn't mean that the ones that are too short have not found applications. In fact, there are some interesting, exciting applications. For example, in oxygen, you can radio label water, right? So H2O, and then that actually allows you to study perfusion nicely. But the problem is because the half-lives are too short, you really have to time everything perfectly. You have to inject a lot of radioactivity into the patient. So then occupationally, the individuals doing the injection have to be careful because they're injecting not like 10 millicuries, they're injecting like 50, 60, 70 millicuries because, the half-life is so small. You have to get significant activity in there. So it does provide some complications, but it can be done. There are a number of exciting applications with really short half-lives. But for routine clinical practice, and I'm kind of oversimplifying, but for routine clinical practice, if a half-life of something is just 1 or 2 minutes, that poses a number of issues. But there are definitely applications. That's why, people have that's one reason people kind of really like fluorine-18 used in PET imaging, which has a half-life of under 2 hours, right? Technetium, as we mentioned, has 6 hours. So these are the kind of numbers people, but again, like for example, nitrogen, many people use nitrogen using a cyclotron to do cardiac PET imaging. So it's kind of a bit of an oversimplification to say that if the half-life is way too short, you can't use it. And we never really said that. We said ideally you want the half-life not to be too short or too long. I hope that answers the question. So here's just an example. So again, we're often radionuclides or radiopharmaceuticals are injected into the patient, or actually, there are examples like with iodine 131 where you drink it up, right? But there's also this way of doing it, right? Using gases and you're inhaling. Okay. So again, I'm sort of showing, here I'm showing different isotopes, which we already showed before, but I do want to emphasize the fact that you could have the same isotope, but different molecules, right? So you could take different molecules and label them with the same isotope, and they're going to have totally different properties. Of course, the physical half-life will be the same, but the biological half-life and how it behaves in the body is going to be totally different. And the applications could be anything. Could be in oncology, could be in brain imaging, could be in cardiac imaging. So these are just some examples. These are all PET tracers that I mentioned. It's impossible to go through all radiopharmaceuticals. And I will emphasize that point. There's just many, many, many different applications. So we're just giving you a taste of things. Here's an example in oncology, in imaging of cancer. This is the bread and butter of PET imaging, FDG, fluorodeoxyglucose, and very important pharmaceutical that you have to know, fluorodeoxyglucose. You can think of it as radio-labeled sugar, okay? So imagine if you are studying, throughout the body, where does metabolism take place? That's what fluorodeoxyglucose does for you. So first, it's injected, it enters the blood, then it's taken up into tissue, and then it goes through this process called phosphorylation, where FDG, fluorodeoxyglucose, becomes fluorodeoxyglucose 6-phosphate, and it actually gets trapped. So this is on the pathway of metabolism, glycolysis, but it gets trapped here, and it just accumulates. So if you have high metabolism, like in a tumor, it just keeps accumulating more and more and more and more because it's kind of stuck in this kind of chemical form. And it just accumulates there. Now, the rest of the organs could also have very high glucose, like in the brain. The brain lights up in PET imaging, right? So in fact, originally, fluorodeoxyglucose, it wasn't used for cancer. Originally, people wanted to use it to study the brain. And they did, and they continue to use it to study the brain. But then later they realized, oh, wait a minute, actually tumors take it up too. And so it starts to go down in other organs, whereas it starts to accumulate in, for example, tumor. So that's why, you know, a patient in the clinic tends to wait for about 1 hour when they're undergoing PET imaging. So they're injected time 0, but then they're imaged at time 60. That's why we have waiting rooms. So you inject a patient in the waiting room, they wait, you know, they're there, and then at around 1 hour, we do imaging. And the idea is by 1 hour, you know, some of the normal uptake has gone down, and you've got, you know, high contrast, for example, in the tumors. Right, so this is one of the processes. In brain imaging, again, just so many exciting applications. You know, we have over 100 neurotransmitters in the brain, and each neurotransmitter, you know, can be associated with different kind of things that transport it, like synaptic vesicles. You've got the receptors that receive it, and then the transporter that takes it back, right? So for example, for dopamine, a very, very important sort of, you know, the dopaminergic pathway and system is so important for the study of many things. The study of, for example, addiction. So you could be targeting, for example, the vesicular monoamine transporter using a particular radiopharmaceutical. In fact, there is such a thing. Or you could be targeting the receptors, the D2 receptors, using another kind of radiopharmaceutical, raclopride. Or you could be targeting the dopamine transporter using something else, methylphenidate or whatever. Okay, so there's so many different pathways and so many different targets that you could be targeting, and imagine so many different neurotransmitters. So there's just so many exciting applications in the brain for us to study. For example, exactly, you know, PET imaging was the modality that was used extensively to show that addiction has an associated physical phenomena in the brain, that the brain mechanism of a person who is addicted has been modified. So this was very important. Again, just showing you some examples. This was at a time when I was at Johns Hopkins University, just looking at the broad range of applications that, for example, we would do. So this is a radiopharmaceutical that is a raclopride that is targeting, for example, the dopamine transporters. Or you could have, as I mentioned, methylphenidate that targets the dopamine transporter. You could be targeting the vesicular monoamine transporter. You could be looking at the serotonergic system. You could be looking at the mu-opioid receptors, the cannabinoid receptor, the so-called marijuana receptor. You could be looking at so many different parts of the brain and functions of the brain. Now, this kind of a image that you see here is because we have accumulated multiple patients, and they've all been registered to the same spaces. Therefore, you know, we're having this question about the detail and the resolution. This is actually because there's many patients mapped to the same space. So that's why we get this really high resolution and low noise, essentially. And not the resolution, you kind of see this, but the fact that we're having low noise while having high resolution is because of that. Cardiac applications. We're going to talk about cardiac applications later, especially when we talk about kinetic modeling. But again, the idea is you could be looking at many different functions of the heart. For example, myocardial perfusion imaging is very, very important in order to study, for example, coronary artery disease, right? You know, how is blood flow in your coronary arteries, which is obviously very important. And if you know, this is one major application of SPECT imaging, single photon SPECT imaging in routine clinical practice, to look at the heart and look at function of the heart. So, and yeah, we do have a question about what was K1, K2, and K3 in that figure that we had for FDG. These are rates, and we're going to talk about, this is called compartmental modeling. Sorry, let me show you this. This is called compartmental modeling, where we think of a radio pharmaceutical in different stages. This could be a physical stages, or it could be chemical stages. And if in the field of kinetic modeling, which we will talk about in later lectures, you know, this kind of compartmental modeling is done, and these are rates. So you would write differential equations. You know, rate K1 is the rate by which you go from the blood into tissue. K2 is the rate by which you go back, and K3 is, for example, the rate by which you go here. And K4 is typically assumed to be 0 because you sort of get trapped here. So this is a kind of, you know, biological, mathematical modeling of the biology that is happening. So therefore, the field of kinetic modeling, and we ideally like to estimate these rates. So we'll talk about those in kinetic modeling.